My name is Helen Kennedy. I'm a PhD student from the University of New England. Our university campus is in a small town in rural Australia called Armidale. Armidale resides on the traditional country of the Anouan, to whom I would like to pay my respects today. The primary aim of my PhD is to revise the taxonomy of a group of shrubs which are endemic to Australia called Melichrus. To achieve this, I am taking an integrative taxonomic approach where I consider both morphological and molecular data for species delimitation. I'm about 2/3 the way through my PhD now, so I have some results. I'd love to take this opportunity to share some of those results with you today. And I suspect by the end of my presentation, all of you will likely know more about this little genus of Aussie shrubs than just about anyone else. So enjoy. Melichrus is in the family Ericaceae. Many of you might be more familiar with Northern Hemisphere heathers like Erica and Kaluna, but Melichrus are in a different subfamily, the Epacroideae, for which Australia is a centre of diversity. Now, Melichrus have a widespread distribution in mainland Eastern Australia and a more constrained presence in arid Western Australia, as indicated in the map in red. The eastern and western groups of Melichrus have been shown to be sister groups through molecular analysis. And my revision is focused on the eastern Melichrus, as that is where the majority of taxonomic questions are. I've included the western taxa in some analyses, but for the purpose of this presentation, I will be talking exclusively about the eastern Melichrus. In Eastern Australia, Melichrus are part of many ecological communities from the coast to Midwestern districts in all three eastern mainland states. You'll find them on nutrient poor and often acid soils in grassy and shrubby woodlands, and especially on the thin soils of outcropping granite, sandstone and trachyte. So this slide is something of an honour roll for Melichrus taxonomy, representing over 200 years of work on the genus. Below the image of each author is a full list of species they recognised in Eastern Australia. And despite the apparently small number of species in the genus, there has been very little agreement between any of the treatments regarding species delimitation. Deserving particular mention is Melichrus urceolatus. This taxon has seen numerous taxonomic changes, and as you will see soon, we are just starting now to understand the structure behind this morphological diversity. The most recent revision and current taxonomy for Melichrus was completed by Betsy Jakes, when she was also incidentally a student at the University of New England back in 1957. Jakes recognised 4 species in her revision. She examined over 200 specimens and made numerous collections herself, and this was a massive improvement on the handful of vouchers referenced by her predecessors. But she had very limited access to type material. Consequently, the revision makes some nomenclatural mistakes. She also noted in her revision that the species as she saw them were generally morphologically variable across their ranges. It has been a key task of my PhD to rectify these nomenclatural mistakes and to test this morphological variability for taxonomic signal. So in the latter half of the last century, 2 new species of Melichrus were collected for the first time, a few hours drive north of Sydney. They were both listed as endangered in the 1990s, but without being validly published. In 2019, I completed the field work needed to name and describe these species. 6 months later, by the time I had published the paper naming Melichrus hirsutus and Melichrus Gbrigii, almost all known plants of Melichrus Gbrigii had been incinerated in the catastrophic fires of the 2019-2020 bushfire season. Melichrus Gbrigii has since been upgraded to critically endangered, and its future is uncertain. I think the story of Melichrus Gbrigii exemplifies why taxonomic studies like my PhD are essential to conservation and need to happen sooner rather than later. And with this in mind, I have set out to revise the taxonomy of Melichrus. It is the first and most fundamental aim of my PhD to test and set species of Melichrus in Eastern Australia. And while I have some other aims for my PhD, this is what I will be focusing on today. Throughout the first 2 years of my PhD, I devoted considerable time to visiting herbaria and to collecting in the field plant material required to test species delimitation. During this time, I also hypothesized a new taxonomic structure for Melichrus, which I expressed as phrase-named putative species. I then used the material that I had collected to test these hypotheses against the existing taxonomy using both morphological and molecular analyses. Today, I would like to share with you some of the results from the molecular analysis. So this slide summarizes the sampling strategy used in this study. On the right is an example photograph of the 6 accepted species of Melichrus in the current taxonomy. And on the left is a map showing all the populations sampled for this study. I used a proprietary reduced representation sequencing service similar to RADseq called DartSeq to generate a data set of genome-wide single nucleotide polymorphism markers, SNPs for short, in an extensive sample set of Melichrus. Over 548 individual samples from 110 populations returned over 80,000 SNPs. On the next slide, I'm going to show you a Pca plot, which was generated to answer the question, are there 6 species of Melichrus in Eastern Australia? Before generating this Pca plot, the full data set was stringently filtered to remove SNPs with a low repeatability score or more than 5% missing data, and to remove alleles which were potentially influenced by linkage disequilibrium or which had a very low frequency. A few individual samples were also removed because they had more than 10% missing data. This left us with a very reasonable data set of 2,000 SNPs, representing all Eastern Melichrus. In this plot, individual plants are represented by single points, and to keep things simple, they are coloured by their identification under the current accepted taxonomy, as you saw on the previous slide. Now, this Pca is rich with taxonomic information, but perhaps most strikingly is Melichrus urceolatus, shown here in green, actually separates into at least 4 distinct groups, which occupy the extremities of genetic variation in the genus. These groups agree with species boundaries hypothesized early in the study based on key morphological differences. I'll give you now just a brief rundown of the putative phrase name species which the circle groups correspond with. At the top, we have Melichrus I. Lagorge and Melichrus tara. These phrase name taxa were coined by a Queensland botanist over a decade ago. I hypothesized that these 2 entities were actually conspecifics based on their morphological similarity, and the molecular results appear to concur. The cluster below that the cluster below that corresponds with a phrase name taxa, Melichrus R.Br, which was segregated from Melichrus urceolatus a few years ago by one of my supervisors. Melichrus R.Br is particularly interesting as some early results from the population statistics suggest it could actually be an allopolyploid. And at the bottom right is a cluster which corresponds with a putative new species, Melichrus Canberra, which I segregated on the basis that it's vegetative and some of its floral characters were more similar to Melichrus erubescens than to Melichrus urceolatus sens strict. It is seen here clustering more closely to Melichrus erubescens than to its current conspecifics. Further subsetting of this part of the Pca does decisively separate Melichrus Canberra as distinct from Melichrus erubescens. The last cluster contains samples representing the sens strict interpretation of Melichrus urceolatus. It clusters more closely to Melichrus procumbens and Melichrus adpressus than it does to its other supposed conspecifics. Subsetting for just this part of the Pca, does reveal three very distinct groups corresponding to Melichrus procumbens, Melichrus urceolatus, and Melichrus edpressus. And further subsetting allows for greater resolution of genetic structuring at the species and population level and provides evidence for several more putative new species in each of these groups. I would love to talk a little more about this just now, but I think I should better move on. In summary, I think there is clearly mounting genetic and morphological evidence for a taxonomic classification of Eastern Melichrus with more than 6 species. So what's next? Well, for the DART-seq molecular dataset, I certainly have not exhausted its analytical potential as yet. I'm in the process of running a construct analysis, which should help me to dissect any influence of isolation by distance in some sections of the dataset, as well as a tree mix analysis to look at gene flow. I'm also looking to infer phylogeny from molecular data. Not only do I have the SNPs and the raw sequence reads from the DART-seq to do this, but I also have another data set on the way, generated using the Angiosperms 353 bait set in a target capture experiment, which includes samples from many related genera to Melichrus as part of a big collaborative project to contribute to the Australian Angiosperm Tree of Life. The results from the phylogeny should provide valuable insight for species delimitation as well. Time permitting, I'm keen to look at the biogeographic history of this genus, which could help reveal the relationship between the arid adapted Western Australian Melichrus and the more broadly adapted Eastern species. All of this is working towards producing the first Flora of Australia profile for Melichrus, which will make this taxonomic classification accessible and usable for a wide range of stakeholders. I'd like to finish up here for today by thanking the wonderful group of people who are part of this project and all the organisations which provide essential support. So thanks very much.